All right, next up we have Ann Martin, who is one of our genetic counselors, and she's going to talk about genetics, the registry, and your family.
All right, great. Can you all hear me?
Yep, you sound great, Ann.
Okay, perfect. Thank you. Thank you, Rachel, and thank you, Gretchen. It was fun to hear about that. I don't know about all of you, but I would like some hot cider right about now. So I, as Rachel said, I'm a genetic counselor with PPMD, and so I am going to be talking with you about genetics and how that impacts your family, and information about our free genetic testing program, and information about our registry. Oops, let me see here. Okay, so I just went through this. This is going to be a bit of a kind of Duchenne and Becker 101, so bear with me, and I appreciate all of you joining on a Saturday morning to learn more about all of this. And I do wish we could be in person, but I think this is a great alternative. As I said, I'll go over genetics and the importance of our genetic testing program, and then get into some information about the registry, and then hopefully have some time to answer your questions at the very end. So what is Duchenne? I feel like this slide is probably not needed for this audience. I'm sure all of you are very familiar with Duchenne, but in case we have any families joining who are brand new to the community, I think it can help to just start with the very basics. So both Duchenne and Becker are muscular dystrophies, and they're both caused by changes within the same gene, the dystrophin gene or the DMD gene. And because of those changes that occur in the gene, that protein, the dystrophin protein, is not made properly. And for that reason, the muscles then cannot function or repair themselves as they should. This diagram here looks a bit complicated, but I just wanted to go over this briefly. The colorful diagram there on the left is showing a protein complex within the muscle cell membrane, and that's actually called the dystrophin glycoprotein complex. And the whole purpose of that complex is really to keep the muscle cell membrane stable, basically to maintain its integrity, because our muscle cells are working all the time. They're constantly going through cycles of contraction and relaxation, and that causes a lot of stress on the muscle cells. And the whole point of this complex and of the dystrophin protein is just to maintain the stability of our muscle cells. So the dystrophin is actually that yellow beaded necklace that you're seeing there towards the bottom. But if that is missing or altered, or if any of the other proteins in that complex are missing, each of those can lead to a different form of muscular dystrophy. But in this case, we're focused on that on the dystrophin protein, because that is what leads to Duchenne and Becker. And the picture on the right there is just another way to think of dystrophin. That's a tire and a shock absorber. And I think a lot of people like to think of dystrophin as being like the shock absorber for the muscle cells. It's there to absorb the stress caused from the movement and the contraction of our cells. So when it's not there, that's why the muscles become damaged. This picture is actually showing what the muscle tissue looks like if we could look at it under a microscope. The diagram, or the photo to the left, is showing what healthy muscle tissue looks like when you look through the microscope and you stain for the dystrophin protein. And you can see that red kind of ring around each muscle cell is showing you the dystrophin. And that's how the muscle should look typically. But the picture to the bottom right is showing the muscle tissue of someone with Duchenne, and you can see there's no dystrophin protein present. So the muscle tissue does not look as it should. And in fact, there's a lot of spaces between the muscle cells, and that's where over time, scar tissue and fat starts to replace some of those muscle cells. So you're going to hear us talking a lot about Duchenne being a disease of the muscles, but it actually is a disease that affects every system in the human body. And our expert care panel that's speaking after me is going to go into a lot more detail about how Duchenne affects all of the different body systems, but it is important to realize it doesn't affect just the muscles. It affects the bones within our skeletal system. It affects the heart. As you know, the heart is a muscle, so a lot of children and adults with Duchenne and Becker and even carrier females can have cardiac complications. There on the bottom, it's showing the respiratory system. Our lungs can be affected because the diaphragm is a muscle. Our digestive tract can be affected. Even the brain, there are levels of dystrophin that are expressed in the brain, and that we think that could lead to some of the more behavioral or learning issues we see in some children with Duchenne. And that last picture on the bottom right is showing the endocrine system, and I'm sure as many of you know, the growth of children with Duchenne is affected as well. So again, our care panel, speaking after me, is going to focus on some of these other systems in the body. And I, as I said, since I'm a genetic counselor for this talk, I'm going to be speaking more about genetics because that's what I love. So bear with me here, but I do think it's really important to have a basic understanding of genetic terminology because you hear so many of these terms thrown around, and especially if you're new to this community or you have a new diagnosis, it can be really confusing. So this is going to feel like high school biology, but I'm going to review this quickly. This picture is showing a cell, a human cell, and of course our body is made up of millions of cells, and in the center of each cell is the nucleus, and within the nucleus, that is where all of the chromosomes are housed, and the chromosomes contain all of our genetic material. We inherit them from our mother and father. If we could actually unravel a chromosome and see that DNA molecule, that is what it would look like. I'm sure you all heard of DNA. Basically, each small segment of DNA is a gene, and each gene codes for something in particular in our body. So in the case of Duchenne and Becker, we're talking about the dystrophin gene or the DMD gene. If we were to even unravel the DNA further, we would be able to see the DNA bases, which are 4 letters, the A's, T's, G's, and C's. And some of you may have seen these letters on your child's genetic test report. I'll go into more detail about that in a minute. But just to kind of give you the basic overview of genetic terminology. So if we were to take all of the chromosomes within a single human cell and line them up, we would see that there's 23 pairs or 46 total. That 23rd pair is the sex chromosome pair, and women always have 2 X chromosomes, and men have 1 X and a Y. And this is really important for Duchenne and Becker because the gene, the dystrophin gene that causes Duchenne and Becker, is on the X chromosome. So it's an X-linked condition. And that's really important to know in terms of the inheritance pattern of Duchenne. And that's why women are typically just carriers, because if we have 1 X with a dystrophin gene change, it usually doesn't matter because we have a second X that works as a backup copy for us. Whereas men, since they only have 1 X chromosome, whatever they inherit on that is going to be expressed. And we'll get into more detail about all of that in a moment. So back to the actual dystrophin gene on the X chromosome, the DMD gene, it's actually one of the largest genes in the human body. It's huge. It contains 79 exons. If you've not heard of an exon. An exon is just a small segment within the gene that contains important coding information. There's some genes that only have a few exons, but the dystrophin gene is remarkable in that it has 79. So it is a large, large gene. And the segments in between the exons are called the introns. In the past, we thought that was really just junk DNA because the introns are spliced out or cut out of the final product. We now know that's not entirely true. They're not completely junk, but I'm not going to get into a lot of details about that. But the point of this slide is just to show you that all of those exons need to line up just perfectly in order to form the correct dystrophin protein. So the introns are spliced out, and all 79 exons have to line up just perfectly in order to have a functional dystrophin protein. Many of you have probably seen this diagram. This is showing all 79 of the exons and how they fit together. It's like a puzzle, really. They have to fit together just so. So if any of those exons are missing or repeated, it can really cause a lot of problems in terms of how that protein functions. So because the gene is so large, there's a lot of different changes that can occur in the gene. There's actually thousands of different changes that can occur within the length of the gene. You're going to hear these terms a lot, like genetic change or mutation or variant. Those all basically mean the same thing. It's a change within the gene. Variant tends to be used more often now. So if you have a recent genetic report, it likely uses the word variant. And you'll also see words such as pathogenic variant. Pathogenic just means it's a disease-causing gene change. It's been proven or shown to cause disease, versus a benign variant. Benign, meaning that it's benign, it does not cause disease. Occasionally, you'll see, this doesn't happen much on Duchenne and Becker reports, but occasionally you'll see something called a variant of unknown significance. That's basically, we don't know if the variant, there's not enough research to know if the variant is truly pathogenic or disease-causing. But certainly, if you have any terms like that on your genetic test report that you don't understand, please send it to us. The genetic counselors at PPMD, we're more than happy to review them. So the table on this slide is basically showing the most common changes that we see within the dystrophin gene. By far the most common are large deletions. That's when an entire exon or several exons are missing from the gene. That happens in about 2/3 of Duchenne cases and even more Becker cases. Duplications happen about 10% of the time. That's when an entire exon is repeated or duplicated. And then the third category are the smaller point mutations. This happens about 15 to 30% of the time. And the point mutation means it's not actually at the exon level, but it's a very small change within the exon. So this is at that DNA base level with the A's, T's, G's, and C's. So this next slide, I think, is helpful to kind of describe these different types of mutations. If you look at the top sentence there, you can think of that sentence as a gene. The old dog ran too far. If you have a deletion, then you have a word within that sentence or an exon missing. Whereas a duplication, you would have an exon or a word actually repeated. And then the third type of mutation listed there is a nonsense mutation. The nonsense mutations are actually the most common point mutations. So again, these are the very small mutations within an exon. A nonsense mutation just means that there is a change of one or more letters in the DNA code, and that creates what we call a premature stop in the gene. So literally, production of the protein from that gene just stops, and you end up with a very short or truncated protein that basically is degraded in the cell and can't function. The other two types of smaller mutations listed there are a missense mutation. That's when just one letter is changed. So in this case, instead of the old dog ran too far, you get the old dog ran too fat, which doesn't make sense. What this does to the protein, usually it means that a different amino acid is inserted in that position, and so the protein functions differently most of the time. And an insertion is just how it sounds. There's usually extra letters inserted within the gene that can also cause difficulties. But by far, what we most often see in Duchenne is either a deletion, duplication, or nonsense mutation. And again, if you have questions about your child's genetic test report or your own report, please send them to us. I'll have our information at the end of this talk. We have 3 genetic counselors at PPMD. We are happy. We actually like to review genetic test reports, so please send them to us, and we are more than happy to talk with you about what that means for you and your family. I did want to quickly go through the reading frame rule, because I think that's really important to understand. If your child has a deletion, you've probably heard the term out-of-frame deletion versus in-frame deletion, and that can be somewhat confusing. So the next few slides, I think, do a good job of trying to show you what that means. Again, I'm going to go back to the sentence. If you have the old dog ran, and that's a gene, and each of those words is an exon, what is supposed to happen is those all line up perfectly and you get a protein or a sentence that makes perfect sense. So that's what typically happens. In Duchenne, if you have a deletion of an exon, then you end up with a protein that doesn't make any sense because the remaining exons basically can't fit together properly. That reading frame has been altered, and that's called an out-of-frame deletion. And in this case, you get a protein that just doesn't work. With Becker, what happens, you can have a deletion, but it's an in-frame deletion because the resulting exons still fit together and that resulting sentence or protein still makes sense. The dog ran. That still makes sense. It's not perfect, but you're going to get some protein made. So that's the difference between out-of-frame and in-frame. I mean, typically an out-of-frame deletion will result in no dystrophin production, and that results in the more severe Becker presentation, typically. Whereas an in-frame deletion, you're going to get some dystrophin protein made that's partially functional, and so those individuals typically have the more mild Becker presentation. This is not perfect 100% of the time. There are exceptions, so just be aware of that. But the other reason why I really wanted to bring up the reading frame rule is this is really the premise for exon skipping. We hear a lot about exon skipping now, a lot of excitement about it. But the whole premise of exon skipping is to turn an out-of-frame deletion into an in-frame deletion with the hope that you will help a child who's kind of programmed to be more Duchenne-like to hopefully be more mild, to have a more mild Becker presentation. I'm not going to go into a lot more detail about exon skipping because I know there's a couple industry presentations that have been saved on our website for all of you, and I think those links were emailed to you, but I definitely encourage you to listen to the Sarepta Therapeutics talk and the NS Pharma talk. Both of those companies work in exon skipping and have talks that I'm sure will do a great job explaining exon skipping even further for you. So I'm going to go back a little bit to the chromosomes and the X-linked inheritance because I do want to touch upon the carrier information, which is so important. So if you are a female carrier, so that would be the woman here on the top right of this diagram. So she has one X that has the white mark on it, which is showing the Duchenne mutation, and then she has that second X with the normal functioning copy. So every time she has a child, there's going to be a 50% chance that she passes on the X with the Duchenne mutation, because she's either going to pass on a normal functioning X or the other X with the Duchenne mutation. So basically, each time a carrier mother would conceive a boy, there'd be a 50% chance he would be affected because, again, whatever boys get on that X from their mother is going to be expressed because they get a Y from their dad, which is what makes them male, but they only have that one X from mom. Whereas if a carrier mom has a daughter, there'd be a 50% chance that she would be a carrier because daughters get a second X from their father. So again, if there's any questions about that, please reach out to us. But one thing that I think is so important to understand, I think there's a lot of misperception out there about who's a carrier. I think there's some moms who think if they have a child with Duchenne, they're automatically a carrier, and that's not the case. Now, if you have a family history of Duchenne, so if you've had a child affected and then you actually have a brother with Duchenne or a maternal uncle, then you would be a carrier. You're what we would call an obligate carrier. We know you're a carrier based on that family history. But in the families where you have a brand new child diagnosed and he's the first in the family diagnosed, the chance that the mom is a carrier is around 70% or 2/3. There's a 1/3 chance that the mom is actually not a carrier and that gene change started new in her child. And it's really important to know if you're a carrier, not only for family planning reasons, and so we can determine the risk of recurrence in the family and the impact on other family members, but it's really important for women to know this for their own health because we know that some carriers can have some muscle symptoms related to being a carrier. The majority of carriers are fine and don't know that they're a carrier, but there are cases where a woman can even be what we call a true manifesting carrier and have more significant symptoms. I think we're trying to move away from that term manifesting carrier, so you may hear female with dystrophinopathy instead. But it's so important to know if you're a carrier. Because carriers are at risk to have cardiac complications, we would want anyone who's found to be a carrier to have a cardiology evaluation as soon as possible. And I'm going to get into a bit more information about how we can make that carrier testing happen in a moment. So I've been talking a lot about genetics and why it's so important. I think we all agree that it's, really the gold standard for the diagnosis. In the past, boys were often diagnosed with Duchenne or Becker just based on a CK level or muscle biopsy, but now genetic testing really has to be done. It is the gold standard. And of course, it's so important for family for the reasons we've been talking about, because we need to be able to offer carrier testing to the mom and then to other extended family members as well. But just as important is the research and the treatment possibilities that are now mutation specific. So it's so important to know your child's mutation and really understand that, because so many of the therapies that are now even approved, but even more in the pipeline, are mutation specific. And again, I'm not going to go into a lot of detail here because we have a lot of these great industry presentations saved for you to listen to, but this slide just reviews some of the genetic approaches to therapies. There is a nonsense mutation read-through therapy that was produced by Sarepta Therapeutics. It's called Adalurin or Translarna. That's not yet approved in the U.S., but it is in other countries. So if your child has a nonsense mutation, I encourage you to listen to the PTC Therapeutics talk. I've already touched upon some information about exon skipping, but it's very exciting that Sarepta Therapeutics now has 2 approved products for exon skipping. They have Exondys 51 for 51 skipping and Vyondys 53 for 53 skipping. Again, if you don't know if your child has the right mutations for those, please reach out to us. We can review the report and let you know. And then, of course, we've all been hearing a lot about gene therapy. There's a lot of excitement and hope about that genetic approach to changing Duchenne. And there are going to be 2 trials, bigger trials that we know of, recruiting later this year. So that is very exciting. Definitely stay tuned, with PPMD through their website for various updates on those. Another thing I want to point out quickly is I know there's a lot of talk about, knowing your right mutation and so many of these therapies being mutation specific, but I also want to emphasize there's a lot of therapies in the pipeline, a lot of really exciting and promising therapies that are not mutation specific. So they would apply to all children with Duchenne regardless of your mutation. So don't feel like if your child doesn't qualify for exon 51 skipping that you're missing out on everything, because that's not the case. So there's a lot of other great industry presentations that are saved on our site that you can listen to. So back to what we as the genetic counselors at PPMD can offer you. We are involved in several different programs within PPMD. I direct the registry, and I'll talk about that in a minute. We also oversee a large pilot program for newborn screening up in New York State, and that's well underway. And then another program that we oversee is the Decode Duchenne free genetic testing program. So I'm going to talk about that quickly. This program has been in existence since 2013. So we have been doing free testing for quite a while now, and it's all possible from support that we get from Sarepta Therapeutics and PTC Therapeutics. So I really want to thank our sponsors for that. But this is really a great opportunity to obtain free testing, and I encourage everyone to take advantage of it. This is not only for a child with a new diagnosis who needs a genetic test done. Of course, it's for that, but we also do a lot of repeat testing for children who had their testing done many years ago or maybe never even had genetic testing. If you have an older report and you're not sure if it's still the best method of testing, we're happy to review that for you and let you know if a repeat test would be beneficial. But very exciting. Over the past year or two, we've been able to include free carrier testing as part of this program, which I think has been so beneficial because historically carriers or women have had a hard time getting carrier testing. So we can now do that for free through our program, which is a really great benefit. All of our testing is done through Perkin-Elmer, and we can do testing on saliva samples as well as blood. So it's we try to make it as easy as possible. We've tested over, I think this is over 1,200 now, patients total through the Decode program, and an additional 200 to 300 carriers have been tested. So if you're interested in this program, please reach out to me. I'll have our information at the end of this presentation. The testing does need to be initiated by a local doctor, but it's fairly simple. They can go to our website, complete a short online application form, and then we help to coordinate all of that testing for you. So definitely encourage you to take advantage of this program. So finally, I want to talk about the registry, the Duchenne registry. As I said, I'm the director of the registry. The registry's actually been in existence since 2007. That's when we started collecting data. It was started with a big joint effort with PPMD and the CDC and NIH and Emory Genetics. It was really a joint effort to start a large patient report registry that would be focused solely on Duchenne and Becker. We now have over 5,000 registrants since we've launched. It's an international registry, so we actually have over 100 countries represented. We have a wealth of longitudinal data, especially on those registrants that have been with us since we launched in 2007. And most importantly, we have recruited or been involved with recruitment for over 100 different clinical trials and research studies. And that's really a great benefit for the families. If you're in the registry and your child's account is up to date, you will get notified when there is a trial that you or your child appear to match criteria for, because the majority of researchers and trial sponsors do come to us to recruit for their studies. So we search the registry to see who appears to match inclusion criteria and then email you to let you know that, hey, you or your child appear to match criteria, and this is who you contact for more information. So it's really a nice benefit. It's great that we have so many trials in the Duchenne space right now, but it can also be overwhelming for families and really hard to keep up with. So the biggest news with the registry recently has been our new app platform. We just transitioned to a new app last fall. We were really wanting to make the registry more user-friendly and just easier for families to access. And since everyone lives on their smartphones these days, we thought transitioning to an app platform would be a great way to keep people engaged in the registry. So if you haven't yet downloaded the registry app, I encourage you to do so. It's very simple. You'll see some welcome screens that describe the purpose of the registry. You click Join Study. You will be taken to our consent. So even if you were in our registry in the past, in our web platform, you do need to reconsent in the app, but I promise it's very simple. You swipe through the consent screens and enter your name and actually sign directly on your phone. And that is how you provide consent. And then you'll be taken to this activities or dashboard screen where you'll see the list of surveys that we ask you to complete. And this is how we collect the core data within the registry. And this is the data that the researchers are so interested in. So I really encourage you to please complete these registry surveys. And if you were in our registry in the past, these should look very familiar to you. They're actually pretty much the same surveys. We did add a new one on top called About Me, and that's where we collect a lot of basic demographics, but that's actually really important to complete. So please make sure you complete that top survey first. As you swipe through the surveys, like I said, it moves really quickly. You just click on the survey. There is one question per screen, so it moves really easily. You just click the correct response, and there's skip logic built in, so you should only see questions that are appropriate for your child. So we really tried to make this as family-friendly as possible. So why should you join the registry? I think maybe some of you have heard this term a lot lately about being a citizen scientist, and this is really a great way to be a citizen scientist. You can really help to advance research and kind of move the needle in Duchenne by just answering some surveys from the comfort of your own home. So it's a great way to really be involved, and you don't have to join a time-consuming clinical trial or research study. Just by joining the registry, you're doing, making a tremendous effort. The data within the registry is really well utilized by researchers around the world, so don't think that you're entering data and nobody's looking at it. It's actually being exported and used by a lot of researchers around the world. And of course, all the data we share is de-identified. So if you have any questions about that, I'm happy to answer. And all of that information is within our consent too. So really just encouraging you to please join the registry if you haven't already. I did want to point out quickly before I conclude that if you go to the App Store, so either the Apple App Store or the Google Play Store, and you download the registry app, you will need this activation code to proceed. If you are in our registry in the past, this was emailed to you back in November when we launched. If you cannot find that email, which I understand, reach out to us and we can have it resent to you. If you're brand new to the registry and you're not in the registry yet, you just need to start at our website. So just go to the DuchenneRegistry.org or connect to the registry through the PPMD site. You click join, you enter your email under this pre-registration screen, and then you will get an invitation from Thread Research. Thread Research is the company who's developed our app. But that email will contain a link to download the app and your personal activation code. Okay, so I went through a lot of information quickly. If you have any questions, please reach out to us. This email here, the coordinator at parentprojectmd.org, that goes directly to the genetic counselor team. We are happy to answer all of your questions. If you want to email me directly, my email is just Ann, A-N-N, at parentprojectmd.org. And then that 888 number goes to our registry team as well. So please reach out to us if we can help with any questions you have about genetic testing or genetic counseling questions or registry questions. We're happy to help.